Hello everyone, I am Dr. Pooja, your biochemistry educator, and today I will be discussing some of the questions which are likely to be asked for your AIIMS MSc Biochemistry entrance examination, as this exam is going to be conducted on 15th of June of this month. So let's start up with the discussion of the questions. Now see question number 1. Rancidity of butter is prevented by the addition of. Now first of all, you should understand by the term what is this rancidity. Rancid means when, and butter is what? Butter is fat. So when fat starts giving a foul smell, this is actually, it happens, this process, rancidity occurs when the fats are exposed to oxygen. So there has to be something, which is preventing against oxidation. So obviously we have to pick an antioxidant. So if you look at all the 4 options, vitamin A, D, E, and folic acid. Folic acid I can write it as vitamin B9 also. So if we compare A, D, and E are fat-soluble vitamins, and folic acid or vitamin B9 is a water-soluble vitamin. But here the question is altogether different. We need to search for an antioxidant. And that is the function of vitamin E also. Vitamin D is actually known as a vitamin which is in search of a disease. No specific disorder is actually related to the deficiency of vitamin E, but it is required as a very, very important antioxidant. So as such, no particular disease is caused, but still vitamin E is responsible for causing of so many, it is acting as an antioxidant. So one of the very important functions is it prevents the rancidity of the fats. And how it prevents? By actually degrading, by actually not letting the formation of free radicals. And that is what is the function of an antioxidant. So the answer to this question would be C, vitamin E, right? Moving on to the next question, oxidative stress. Now see, oxidative stress, it is a very, very important word, oxidative stress, which is released due to oxidation. Now, basically what happens is, I won't take this question into that much detail because this is a very, basic question and a very elaborative question also. In our body, oxidative stress is mainly due to the production of excessive free radicals. First of all, let me just tell you the answer to this question. It will be A. Now, what do you mean by the term free radical? These free radicals, they are always in search of an electron, always. This is the definition only. So electrons, we know they are negatively charged. Okay. Now, say this is a free radical. So it in search of an electron, what will happen is, this is the cell membrane. It will start, damaging the cell membrane, or it will start eating the cell membrane. So what happens is, when it starts damaging the cell membrane, it leads to excessive hemolysis in the body. So oxidative stress, the main purpose is how it is caused. It is caused due to the production of excessive free radicals. And all the antioxidants which are there in our body, they function as to prevent the formation of free radicals. There are so many antioxidants which are occurring in our body. Superoxide dismutase is there, glutathione peroxidase is there, E is there, vitamin C is there. There are different chain breaking antioxidants also, right? So here simply oxidative stress is due to the production of excessive free radicals. It has nothing to do with indigestion, nothing to do with BMR also. Moving on to the third question, most of the members of vitamin B complex are primarily used as. Now, first of all, vitamin B complex. Why vitamin B is known as a complex? Because it consists of a series of vitamins like B1 is there, B2 is there, B3 is there, B5, B6, B7, B9, and B12. B1 is thymine, B2 is riboflavin, and always remember this is nicotinic acid. They always work in their active form. जैसे B1, the active form of B1 is BP, B2 is FAD, B3 is NAD or niacin, which is commonly known as, B5 is pentotheniine, or it is coenzyme A. B6 is pyridoxine, and active form is PLP. B7 is biotin, B9 is folic acid, and B12 is cobalamin. Now, very important feature about all the B complex vitamins is that most of them function as coenzymes. Coenzymes work in cooperation with the enzymes. Vitamin D is used as a hormone, but it's a fat-soluble vitamin, so it won't come in this category. Enzymes are proteins, so it will be coenzyme. Like B1 is a very, very important coenzyme for so many important reactions. Like it is a component of pyruvate dehydrogenase complex. If you want to know the detail about these vitamins, I have discussed them in my other videos also. So you can just go through that because vitamins play a very, very important role in our body. And from obviously examination point of view, they are important. Similarly, B6 is there. B6 is responsible for the amount of protein intake in our body. Right? So all of them are functioning as coenzymes. Clear? So the answer is C. Which is of the following is the most important reactive group of glutathione? Now, see, what is this glutathione? It is actually a tripeptide. Tri means 3, peptide means they have a peptide bond. And this glutathione is made of 3 amino acids. And the mnemonic for that is glycine, cysteine, and glutamic acid. Or it is simply GCG. And the function of glutathione is it is used as a reductant. A very important reductant. How? Because of its sulfhydryl group. It's a very important reducing agent. And this sulfhydryl group is SH group. And this SH group comes from cysteine. Because cysteine is a sulfur-containing amino acid. So the reactive group is this sulfhydryl group. And glutathione exists in 2 forms. This is SSG. This is oxidized form of glutathione. And GSH. This is the reduced form of glutathione. So it is clear. SH sulfhydryl group. So the answer is clear. Moving on to the next question. Individuals can have relatively high levels of pyruvate in their blood due to. Now see, vitamins are there, vitamin D is there, thiamine and alcohol intake. Now, if these type of questions are asked, again, just remember what are coenzymes. Because most of the vitamin B complex, they serve as coenzymes. And basically, you should know what is the fate of this pyruvic acid in our body. All of us know that this pyruvate is actually produced from oxidation of glucose by glycolysis. Now, this pyruvate undergoes 2 different fates. Either it will react in the presence of oxygen or it will react in the absence of oxygen. So if it reacts in the absence of oxygen, it is simply lactic acid or lactate fermentation, which happens in the skeletal muscle. But the most important process which takes place in the presence of oxygen in the mitochondria, it forms acetyl coenzyme. And this reaction is strictly irreversible. Now see, when the reaction is reversible, you can say there cannot be any increase in pyruvic acid because whatever lactate is produced, it will be converted back to pyruvic acid. So we come over to irreversible ones. That means we have to concentrate on this reaction in which pyruvate is being converted into acetyl coenzyme A by a irreversible reaction. And here the enzyme is PDH. You know, this PDH stands for pyruvate dehydrogenase complex, and it is an example of multi-enzyme complex. And this enzyme, particularly this reaction, I have discussed in my video so many times. So here I won't take much of your time. I'll just give you the brief introduction of this enzyme. This is a multi-enzyme complex. It has 5 coenzymes to work with it. B1, B2, B3, B5, and lipoic acid, like multivitamin. And the most important one is B1, which is thymine. So obviously, if thymine is absent from the diet, there will be increased concentration of this if B1 is absent or thymine. And that is what the question says. Individual can have relatively high levels of pyruvate in their blood due to thymine deficiency, because it is a component of PDH, an example of multi-enzyme complex. And moving on to the next question, what type of chemical reaction is involved in conversion of isocitrate to alpha-ketoglutarate? Now see, this is a very important reaction. Isocitrate, ICD. Always remember it has 6 carbon atoms because citric acid has 6 carbon atoms, and this is a reaction of TCA. Now ICD is converted into alpha-ketoglutarate, which has 5 carbon atoms. Now just remember, only 1 carbon atom is given out. Now 1 carbon atom is given out by a removal of carbon in the form of carbon dioxide. Now removal of carbon dioxide is known as decarboxylation. D माने removal, किसका removal? Carbon dioxide का removal. Okay, now this reaction is TCA, and all of us know TCA, obviously it is aerobic oxidation. Always it occurs in the presence of oxygen. So the process is oxidative decarboxylation. And this is first reaction of TCA in which 1 molecule of carbon dioxide is given out, and second is given out when alpha-ketoglutarate acid is converted to succinyl coenzyme. So the answer to this question would be B. Clear? The next question, principal regulation point in the biosynthesis of fatty acids is. Now see, biosynthesis of fatty acids I have again discussed in detail in my video. Here, simply, this is known as lipogenesis. Whenever we talk about any process, we should know about the regulatory step. Yeah, rate limiting enzyme. And rate limiting enzyme of lipogenesis is acetyl coenzyme A carboxylase, which undergoes carboxylation, where acetyl coenzyme A combines with 1 more carbon dioxide, because this is carboxylation, and it forms malonyl coenzyme. And one important thing, biotin is required as a coenzyme for carboxylation. This is the rate limiting enzyme of carboxylation. Very, very important. If we take citrate dehydrogenation, no beta lactamase, no ketoacyl. This is the component of fatty acid synthase, but not regulatory. So always remember a rate limiting enzyme of lipogenesis or synthesis of fatty acids is acyl coenzyme A carboxylase, and coenzyme required is biotin. This question can also be asked in the exam. The next question, D-glucose and D-mannose are linked as. Now see, glucose and mannose, and the options are anomers, epimers, optical isomers, and diastereomers. Epimers. Anomers differ at C1. Epimers differ at any other position except C1. Optical isomers are DNL and diastereomers are altogether. The mesotartaric acid is there. Okay. So epimers are the isomers which differ at any other position except C1. So if we talk about glucose and mannose is a C2 epimer of glucose. Right? And galactose is a C4 epimer of, right? Then moving on to question number 9. Collagen is rich in a very, very important connective tissue, collagen. You know, it is synthesized also in the body. And the most important amino acid which is required for the synthesis of collagen are glycine, proline, and lysine. Proline is hydroxylated, so hydroxyl proline is also present. Proline. And lysine is hydroxyl lysine, which is also present. And vitamin C is required for the conversion of proline to hydroxyproline, lysine to hydroxyl. Now here you see the options, glycine, and vitamin C is also required for the synthesis of hydroxyproline to proline, hydroxyl lysine from lysine. And that is why we say deficiency of vitamin C leads to scurvy. Why? Because there is a defective collagen formation. The next question, in SDS-PAGE. Now see, SDS, a very, very important anionic detergent, which is actually depicting the size of the protein. This SDS stands for sodium dodecyl sulfate. Dodecyl sulfate. PAGE stands for polyacrylamide gel electrophoresis. We know that electrophoresis is the most important technique to separate the protein on the basis of size, on the basis of charge, and the principle involved is isometric point. But SDS is the anionic detergent which has a tendency to actually combine with a dipeptide. The most recently used technique. So it will be the size of the protein. Very, very important again. Moving on to the next question. What is the effect of urea and formamide on DNA? Whenever we talk about urea and formamide, they are actually acting as the kind of chemicals which denature DNA, destabilizes DNA. Means they are unwinding the strands of DNA. Now see, what are the options? Obviously, they are going to decrease the TM of DNA. Now, what is this TM? TM is known as the melting temperature of DNA. And TM is actually when half of the DNA strand is denatured. And this becomes the principle also actually whenever we have to separate the DNA strands. Whenever we have to denature the strands, whenever we have to break the bonds, we always measure as per the whether it is hyperchromic, whether it is hyper. So urea is basically added to destabilize or decrease the growth of DNA. And Tm is the melting temperature of DNA when half of the DNA strand is denatured. Lineweaver-Burk plot. Now see, Lineweaver-Burk plot is nothing but it is a reciprocal of Michaelis-Menten equation. Basically, Michaelis-Menten, they were the scientists who gave an equation of steady state kinetics. Steady state kinetics was like this. Like this. Saturation kinetics or zero order kinetics. And their equation was V naught is equal to V max S over Km plus S. So these Lineweaver was the scientist who reciprocated the equation. Reciprocated this equation, came out to be like Km V max S plus over V max like this. So this equation was actually known as the equation of straight line also, where this was the slope and this was the intercept. Y is equal to mx plus, just to calculate the slope and intercept. And that is why it is also known as double reciprocal. The other name of Lineweaver-Burk plot is double reciprocal plot, and it is the reciprocal of Michaelis-Menten equation. So the answer to this is B. Next question. The enzyme responsible for removal of supercoiling. A very important question from examination point of view and a very interesting question. The removal of supercoiling. Always it is topoisomerases. Topoisomerases is a stress relieving enzyme. Because we know that DNA, whenever we have to pull apart DNA, what will happen? It is like a rope. Just to prevent DNA from being denatured, just to prevent the breaking of bonds, we have to put that enzyme which can release the stress which is caused due to unwinding of DNA. Because either it will be supercoiled or it will be undermined. So topoisomerases is a stress relieving enzyme which is actually responsible for removal of supercoiling. So answer is topoisomerases is of two types, topoisomerases 1, topoisomerases 2. 1 cuts only one strand, 2 cuts both the strands, and just to protect them from denaturation. Then primase is which is helping in initiation of DNA replication. DNA polymerase, which is helping in polymerization, and helicases is again which is helping in unwinding of the helix. Because whenever we unwind the helix, it has in the shape of a replication fold. So this helicases, single strand binding proteins, will help in unwinding of helix. But whenever we talk about supercoiling, it is topoisomerases. Then next question, hat medium used for hybridoma production. Now see, hybridoma technology is a very, very important technology which is also used for the selection of monoclonal antibodies, and we always grow them. So this hat medium actually consists of, it stands for hypoxanthine, A stands for aminopterin, and T stands for thymidine. So basically, aminopterin is a competitive inhibitor of dihydrofolate. Right. So it gives a, kind of indication to select the monoclonal antibodies. So it will be thymidine, not thymine. Thymine is B1, whereas thymidine is, because sometimes the get confused, thymine is a pyrimidine. Right. And then the last question, which of the following organism is exploited for transfer of genes in the plant? It's a kind of vector, you can say, in plants. And the most important one is used as a vector in plants is Agrobacterium tumefaciens. Not Staphyl, not Escherichia coli, not Clostridium. So the answer is right. So these questions which I have discussed today, these are very, very important questions, and they will be very helpful for the students who are preparing for their MSc entrance exam, basically for biochemistry as well as for biotechnology. So many students were requesting me just to upload a video to share some of the questions. So this will be of a valuable help for those students. And in fact, this video will be helpful for those students who are preparing for the nursing exam also and paramedical exam also. Okay. So wishing you all the best for your upcoming exams. And I hope whatever questions I discuss with you are clear to you. Thank you so much. Have a great day.